Good morning, dear students of class 8, and I am happy to welcome you all to today's botany class. Today's class is going to be a revision video about chapter 2. What is chapter 2? Reproduction in plants, right? So let's revise this topic for today's class. You need to learn the definition for reproduction. What is reproduction? It is a process which happens only in living organisms and it produces offsprings, that is the child or the younger one of its own kind. How the parent is, similarly only will be the offspring, that is the next generation. And why you need reproduction? Only when there is reproduction, there can be continuity of the species, that is generations can be produced only when there is reproduction. So these all you have to keep in mind. Then there are some modes of reproduction which are sexual or asexual. So any organism can reproduce either sexually or asexually, and in this lesson we are going to learn about asexual reproduction in plants. Apart from plants, there are also other organisms which are neither plants nor animals, and about their reproduction also we are going to revise. Usually reproduction in plants can happen in three ways, called by spore formation, by fragmentation, or by vegetative reproduction, vegetative propagation. So spore formation, fragmentation and vegetative propagation are the three methods by which plants usually multiply. And what about other organisms? They multiply either by budding or by binary fission. All these technical terms you should be familiar with for exam point of view. And how asexual and sexual reproduction differs? There are three differences given in your textbook. In asexual reproduction there is only one parent. In sexual reproduction there are two parents. In asexual reproduction there are no gametes involved, but in sexual reproduction it can happen only by the fusion of the male and the female gamete. And in asexual reproduction, the young one will be just a Xerox copy of the parent. It will have all the features of the parent, but in sexual reproduction, the young one will differ from the parent. Like in human beings, we are not like Xerox copies of our parents, right? We will have some different features which vary from our parents. And this asexual reproduction, as I already told you, there are three methods, no? Spore formation, fragmentation and vegetative propagation. Now we will revise about these three types of asexual reproduction. In spore formation, there is a structure called spore, like a black dot it will appear. Can you see here black dots? By spore formation, there is asexual reproduction mainly in mosses and bread mold. Rhizopus all is there, no? That all reproduces by forming spores. The sporangium will burst out and give out the spores, and when that spores form on the ground, each spore will develop into a new plant or a new species. This is by spore formation, and the spores are globular. Globular means like round or spherical in shape. What is fragmentation? There will be one organism which will be broken down into two or many fragments, and each fragment will have the ability to develop into a new organism. That is called fragmentation. Then what is vegetative propagation? First of all, vegetative propagation can be natural. It may happen by itself or artificially, like man can also induce vegetative propagation in plants. First of all, this word propagation means multiplication. Propagation means multiplication, and so the vegetative parts will multiply. What are vegetative parts in plants? You call three parts as vegetative parts, namely roots, stem and leaves. So if a root is going to multiply, or a stem is going to multiply, or a leaf is going to multiply, then you can call it as vegetative propagation. And from after multiplication, each new part will develop into a new organism. So this is the concept of vegetative propagation, and can be naturally happen, like naturally a root, a stem or a leaf can multiply, or artificially we can force the plant to multiply. How vegetative reproduction happens naturally? See, in root, suppose if there is stored food in a root and the root is swollen in nature, there can be buds formed in a root in that stored food product, which is called as slips. If a bud is formed, it is called as a slip, and each slip will be detached from the root and it will develop into a new plant. This is how it multiplies. What about leaves? In leaves like bryophyllum plant, this is bryophyllum plant, the leaves will develop small buds and each notched with notched margin. See, this is notched margin, like pointed ones. If the margin is there, each notched margin will produce a bud, and that plant, small plant, is called by the word plantlet. See, if a stored root produces a small bud, it is called as slip, and if a leaf produces a small bud, it is called as plantlet. And this plantlet, each plantlet will develop root and shoot and develop into a new plant. This is how it happens naturally. Then how vegetative reproduction happens in a stem? In the stem, it can be either subaerial. Aerial means above, underground means below. What is this subaerial word? Which means a little below the ground, like it is not very deep but just below the soil surface. If it is present, it is called subaerial. How it happens? See, like this, you will have a horizontal stem like structure which is called by the term runners. Okay, so you should not write stories when I ask in the exam. Just these technical terms and keywords I need in your exam paper. Clear? So memorize these words instead of writing stories. And then there will be runner and also roots. See, many roots develop at the buds. Where you can find the buds? It is found in every node. In this method of reproduction, every node will develop bud, and from the bud the roots will emerge, and if you cut and put it under the soil, it can develop into a new plant. This is the process of multiplication here. What about this case? In underground stem, it can be of 4 types called corm, rhizome, bulb and tuber, about which we will be learning in detail in the next slide. So I told you underground stem are 4 types called corm, rhizome, bulb and tuber. No? So for this you have examples for all these that you have to memorize for exam point of view. Example for corm is gladiolus. So this is corm's example. What you can see here, you can find swollen structures, and from this buds will develop, and also you can see a cluster of roots. Like monocots, you will find a bunch of roots in the case of corm, and then every bud will have the ability to develop into a new plant. What about rhizome? Rhizome, here the stem is thick. The stored food is in the form of stem, which is called ginger, and from here the nodes and internodes will develop buds. From every node in a ginger, you can find bud formation, and every bud will produce roots also, and it has the ability to develop into a new ginger plant. What about bulb? Bulb's example is onions. In onions, actually you have fleshy leaves. These are actually modified leaves. This is modified stem and this is modified. Onion is an example for modified leaf, right? So here also apical buds will develop or axial buds will develop. These terms all you have to memorize. Apical buds, which is at the tip, and axial buds will develop, and each bud will develop into a new shoot. And the 4th type, potato, it is a tuber type, and here also this you must have seen at your home all, no? There you can see it is called eye. In all these you call them as buds, and this is called eye where the buds is formed. It is not nodes or internodes. At the eye of every potato the buds are formed, and again if you cut this piece and put it into the soil, it will develop into a new potato plant. And then what is artificial vegetative? So all these what we have seen before, no, they are all happening by natural means. Simply if you place a potato at your home also, it will develop buds from the ice, right? We do not force them, but artificially man-made vegetative propagation methods are there. What are they? What is artificial propagation? First of all, it is man-made. What is the advantage? Many plants can be produced within a short duration. If it has to happen naturally, it will take time. If we force that process, it can happen quickly. and the applications, it is usually used in agriculture and horticulture. Artificial vegetative propagation can either be by traditional methods or by advanced methods. Traditional methods include cutting, layering and grafting. What are traditional methods used for artificial vegetative propagation? Cutting, layering and grafting. And modern method, tissue culture method is an example for modern method. What is cutting? Simply this with rose plant all we do at home. We cut the stem, put it into the soil. After some days, roots will develop as well as shoots, that is the new leaves will develop. This is cutting method. Examples in rose and in sugarcane, you can see this method. What is layering method? Take a branch, just bury under the soil, put a stone on that so that it doesn't pop out, and after some days you can find roots developing here. So this is layering method. Example in jasmine plant all at home, this also we do, right? So example is jasmine. Grafting, here from one plant you cut the upper portion and you fix it to another plant's lower portion, like you cut this and take a upper portion from another plant and join it with this. So here two plants are involved. So the upper portion what you are going to join with the new plant, it is called scion. And what about the lower portion already which is present in the pot? It is called the stalk. So joining of scion and stalk is called grafting, and after some days it will get attached and develop into a hybrid variety of plant. This is called grafting, and you can do this in rose and mango plants. You can apply grafting methods. Advantages and disadvantages, you can just read your text. What about tissue culture method? In tissue culture method, you cut a portion of a plant. It can be a small portion of a leaf. You put it into a nutrient medium, a nutrient source for the plant to develop. After some days, it will develop a structure called as callus, which is a mass of cells, and this callus after some more days will develop into a new plant. So from one small bit of leaf, a new plants can be emerged, and this method is called as tissue culture method. So by this method, within a short duration, you can produce as many small bits of leaves you have, that many plants can be produced in tissue culture method. So it is a very advanced method. And what about sexual reproduction in other organisms? So now we are going to learn about organisms which are neither plants or not animals. How do they multiply? They multiply by three methods called budding, binary fission or multiple fission. What is budding? Budding means a small bud formation. For this, first a portion of this will be swollen. Before that, the typical example for budding is hydra. This also you have to memorize. Apart from that, in aquatic coelenterata also budding can be found. Here a bulb, I told you, swollen structure is formed first, and that is called by the term bulb, and wherever there is a bulb, a bud is formed, and one bud can develop into a new individual. After some days the bud will grow and it will fall down and develop into a new individual. And how a bud is formed? In this, why a bud is formed? Because at some portion of the hydra suddenly there is more multiplication of cells, and that's why this region is swollen and it will pop out as a bud. This also you should know. What about budding in yeast? That was budding in hydra, right? Yeast, what is yeast? First of all, yeast is a fungi, but it is a unicellular fungi. This I have told many times in your classes. So yeast is a unicellular fungi. Here what happens? The yeast will have nucleus. The nucleus is first equally divided into two portions like this, and then the bud will be cut off from the parent, and the bud will develop into a new yeast organism. And binary fission, binary is 2 and fission means like cut or partition. So here also, just like yeast, first nucleus is divided into 2, then the cytoplasmic content is divided into 2, and then the nucleus and the cytoplasm, both of them will individually be separated. So now you have two daughter cells from one parent cell. This is binary fission and it happens in amoeba, and it will happen only when the condition is favorable. When the condition is unfavorable, amoeba usually develop a cyst. Cyst means a thick outer covering. It will form an unfavorable condition. If the condition is favorable, it will opt for binary fission. During unfavorable conditions, it will opt for this multiple fission method, where a cyst or hard covering is covered, and when this is covered, the nucleus will will multiply into many small bits. So many nucleus will have, and on the return of favorable conditions, whenever there is good environment outside, this cyst wall will break or it will burst, and it will release all the nuclei, and every nuclei will have the ability to develop into one new amoeba or a individual, and it is called the daughter cell. Then we will revise about pollination. Pollination means, you should know the definition first, the transfer of pollen grains from the anther to the stigma of another plant, and it always happens within the same species. This definition you have to memorize. There are two types of pollination called self-pollination, which happens within the same plant, and cross-pollination, where the transfer of pollen grains is from one plant, from the anther of one plant to the stigma of a second plant. This is called cross-pollination. And there are three agents of pollination called insects, wind and water. And there are some requirements for every pollination. If animal pollination has to happen, it is supported by birds, insects, butterflies. This list you have to memorize: butterflies, animals and bees. These all can support, and there are certain qualities of flowers to be pollinated by animals. What are those qualities? The flowers should be very big in size, that is, they should be large and showy. They should attract the insects, so they should be beautiful and brightly colored and have a sweet smell. Fragrance should be good. Examples of such pollination is in sunflower and hibiscus. So here the pollen grains will stick to the insects, and the insects, when they go and sit in the stigma of another flower, then pollination will happen. What about pollination by wind? There are some qualities like there should be large number of pollen grains, and the flowers are not brightly colored as we have seen in the previous slide. The flowers are not colored or showy, they are white in color, but they are light in weight, so that they can be carried away by the wind. Also, there is no sweet smell or fragrance or nectar, but the stigma which receives them is always sticky in nature, so that it can trap the ones that is moving about in the wind. And pollination by water means the transfer happens within the water itself. Pollen grains is transferred to the stigma within the water itself. These pollen grains, usually if you see, they will have a waxy coating, so that the pollen grains are not soaked into the water. It's like a protective covering. So pollen grains will float about in the water and reach the stigma, and you can see this in Vallisneria plant. The last topic is about fertilization. So after pollination happens, there is fertilization. So now the pollen grains, after having reached into the stigma, they develop a tube-like structure, and it is called as pollen tube, and this will reach down until it will reach the female gamete. Which is the female gamete? The ovary, and the ovule inside is the female gamete. No? So what you have inside? We have the male gamete, that is the pollen grains, which will pass through the pollen tube and reach the ovary. Then there is fusion, and the male and the female gametes will fuse to form a structure which is called as zygote. Now the zygote will divide rapidly. After and again it will divide, and it will form a structure which is called as embryo. See, generally after fertilization process or pollination process, the ovule, that is which is inside the ovary, you have ovule, no? This ovule is the one which develops into a seed, and ovary is the one which develops into a fruit. So after fertilization, fruits and seeds are produced from ovary and the ovules. So this is a quick revision about the lesson reproduction in plants. I hope you are all learning and preparing well for your exams. So until I meet you in your next revision class, you all take care, keep learning, and bye bye.